Good afternoon. I also want to first thank BGRI 2018 organizers for giving me this opportunity to share my research. As we all know, bread wheat has a very low genetic diversity, and for that matter, breeding for resistance is a major problem. So currently, to overcome this problem, we usually resort to bringing in wild resistant resources from wild relatives, and this comes with problems like linkage drag, and it's also time consuming. So the question is, can't we create resistance directly within wheat itself? And yes, so with this opportunity, I'm going to share with you how we have created resistant germplasm through the modification of MYC4 transcriptional factor gene. From a study of wheat-RAS pathogen interaction, we realized the upregulation of some genes after being the plants, susceptible and resistant plants being inoculated with leaf rust pathogens. And as you can see over here, there was an upregulation of this gene, one of the genes, as I'm showing here, from 3 days post-inoculation, 5 days post-inoculation, and 8 days post-inoculation. This result was interesting because this corresponds to critical time points of RAS disease pathogenesis. So we looked at because this gene, which we later annotated as MYC4, has 3 homologs, 3 copies. We looked at the sequence result of the susceptible sample from 5 days post-inoculation, and we realized that the sequence results correspond to the 1A genome. So in a separate study, we inoculated Opowa, which is also susceptible, with buffer, and then with stem RAS race QFSC, and there was also an upregulation, but in this case, the pattern was different. When we sequence again the result from the samples from 5 DPI, we realized that the 1A and the 1B was what was upregulated. So then, this, together with the previous result, gave us a reason to believe that the upregulation of this particular gene is to benefit the pathogen during its interaction with wheat. So one would think that if this is true, then the reduction in the level of expression of this gene should probably confer some level of resistance to the plant. So to start testing our hypothesis, we employed the knockdown of this gene using barley stripe mosaic virus induced gene silencing. It's a long one, but take it like that. And the results from this shows that there was an enhanced resistance after we silenced the MYC4. Bear in mind that MYC4, as I said, has 3 homologs. So in order to successfully knock out the 3 homologs to avoid probably functional redundancy, we use an insert which is a conserved region for these 3 homologs. So after observing this phenotype, we checked the expression level of the gene in the silenced plants relative to the control, which the gene was not silenced, and we realized about 40% reduction, indicating that this resistance is attributable to the reduction in the level of this gene. So this result encouraged us to create mutation in this gene. So luckily for us, we had cadenza EMS mutagenized population created by Jonin Center, and then we had that from 1A and 1B, but the 1A homolog did not have enough information for us to confirm. So we first confirmed the mutation from the 1A, and we identified two mutants, one being homozygote, one was a change from G to A at position 12, and amino acid change from proline to serine. And then for that matter, we named that mutant as P12X, indicating that that is a proline at position 12, change to serine, and then MYC4 1B homolog. As you can see here, this was a sequence result indicating the homozygosity of this mutation. The second mutation was a heterozygote, and we ID as M17I, which was a mutation from methionine to isoleucine. And as you can see, a double peak also indicating the heterozygosity of this particular mutant. So we proceeded then to test the mutant with stem RAS, race TMLKC, leaf RAS, PBJJG, and stripe RAS. The results was interesting. As expected, we saw that in the case of the stem RAS, there was an enhanced resistance. And then also with the stripe RAS, there was some level of resistance, even though you can see a lot of correlation, but there were hypersensitive responses indicating resistance. But leaf RAS, in this case, has no resistance. And this was not surprising because from the beginning, I indicated that when we inoculated with leaf RAS, the homolog that we identified was only 1A. So in this case, we are using a mutant which was a 1B. Mutant. So this was actually expected, but we are proceeding to test the mutant of 1A with leaf RAS to get a clearer picture of this. So we also check the expression level of the gene in the mutant relative to the wild type. And as you can see, there is no significant difference, which indicates that this is just a loss of function mutation, and so the gene is still being expressed. As I indicated, we had one of the mutants being heterozygotes. So for genetic analysis, we tested the F2 population of this mutant with the same stem RAS race, and we realized a phenotype distribution that was corresponding to the segregation ratio as expected. And here we have the first one was a resistant one, and the middle, like in between, kind of heterozygote, and then this being really very successful. So we are expecting that the sequence result of this would have, the sequence result of, the sequence result of these would have the wild type nucleotide, and then this, the first one, should have the mutant nucleotide to confirm that this mutation is actually what contributes to this resistance. So after this, I think we are all wondering how the mutant look like, because most of these mutations probably comes with some detrimental effect. We were also curious as well, so we looked at the mutant compared with the wild type under greenhouse conditions. At this current point, this is how the mutant look like, but we are proceeding to create near isogenic lines for further testing. Well, we have the material now, but the next question will be why the mutation of this gene confers some resistance to the plant against stem RAS. So to answer that question, we begin to look at what is known and unknown of this gene first. So MYC4 belongs to beta beta helix, basic helix loop. The two blue colors represent the helix structure, alpha helix structures, and the third, the red one being the loop connecting them. And these genes were first identified in human cancer patients. And they have, with this structure, they also have DNA binding domain and a leucine zipper domain. This is the motif that these genes are noted to bind to. This structure helps the gene, one, to bind to other genes and to upregulate or downregulate their expression. It also helps the gene to form homodimers or heterodimers with other DHL-H transcriptional factor genes. What about plant? What do we know about this gene in plant? In Arabidopsis, so far this gene has been identified to be an interactor of JAS domain, and it has been known to do that in connection with MYC2 and MYC3 to regulate JA response genes. We also know that it's also interact with glucosinolate related MYC transcriptional factor genes to regulate the biosynthesis of this molecule, secondary molecule. But the function of this gene in connection with the pathogen interaction with the host is what we do not know. But clearly from this basis, we can understand that this gene probably has something to do with defense response of the plant. So this actually forms the basis for our next research, which we first want to understand the mode of action of this gene. And of course, we are starting by looking at the expression of SA and JA related marker genes in the mutant relative to the wild type. This will give us an indication of which of these pathways this gene is associated with. Then we will proceed to look at the genes that interact with MYC4 in wheat. We are also testing further the mutants with other races, and we also generated a double mutant from the 1A and the 1B, and which we'll also be testing with the pathogens to get a better picture of base. So we are generating near isogenic line also for field trials and yield penalty assessment. So on that note, MYC4 1BL homolog we have identified to have some enhanced, after its mutation, the plant has some level of enhanced resistance to TMLK C stem RAS erase. But most importantly, this study is giving us presenting to us a strategy that we can easily, fast, and effectively identify RAS target genes in wheat itself, mutate them to create resistant germplasms directly within wheat. This would probably overcome the problems associated with bringing in resistant resources from other the wild relatives. On that note, I want to acknowledge my principal supervisor, Dr. Lee Hua, who has been giving direction with this research, and Dr. Hunter, my lab manager, whose previous research served as a basis for this study. Dr. Mike Jeru, my department head, who was instrumental in getting the mutants, and Mina Botros, my lab mate. Of course, NSF, BRED program, and Montana State University. On that note, thank you for your attention. and we're glad to answer any questions.
Thank you so much, Bernard.